Here in the distal convoluted tubule, we're going to see both reabsorption and secretion taking place. Now again, the name distal comes from the fact that we are further away from the glomerulus, right? So proximal to the glomerulus, distal to the glomerulus. And things are pretty different at this point, right? We've actually made a lot of changes to the filtrate. The tubule cells are still simple cuboidal cells, but now we're not near as interested in reabsorption, or it's not near as dominant, and so we really don't see microvilli on these cells. They're just cuboidal cells, right? They give you just like a couple little microvilli there. Now, reabsorption is still taking place here, right? So our blue lines are showing reabsorption. And what we're gonna see is that we have reabsorption of, say, things like sodium. This is a key one to understand. Typically when sodium is reabsorbed, we're actually exchanging it for potassium. And what I will say here too is, instead of just this like bulk reabsorption that happened in the PCT, this is a lot more like fine tuning, and we're definitely going to see our hormones playing a bigger role in deciding what gets reabsorbed. So they're showing you here that aldosterone plays a big role here in actually producing more of these transport proteins, right? This is an aldosterone regulated pump. So there are just some exchange of sodium and potassium, but definitely hormones can help change that as well. So we're reabsorbing sodium, right? In exchange, potassium is secreted. And this will become really important in the next chapter as we start following kind of the balances of some of these electrolytes, right? Again, aldosterone can increase the number of pumps, right? So that we can have increased sodium reabsorption. But that does come at the loss of potassium. Okay, so that's a really important reabsorption that takes place here at the DCT. Of course, water is going to follow salt, sodium, right? And again, our hormones, things like ADH, absolutely regulates the number of water transport proteins that there are, but water movement is always a passive process, right? And it's just following where salt goes. Okay. This is the same picture 3 times. Okay, the other big thing that happens here at the DCT, and we haven't really talked much about this yet, is secretion. Remember, secretion means actually putting things into the filtrate that's going to become urine. So we see a few things being added here. We know, right, we just talked about the fact that potassium is also often secreted in exchange for sodium. Here they're showing us a couple other things. At the DCT, we make a lot of choices that are going to influence pH. Here we actually see hydrogen being exchanged for sodium, and so we absolutely can secrete hydrogen instead of potassium. If we would like to get rid of hydrogen. So if your blood pH is too low, it's too acidic, you can use the kidney to secrete those hydrogens. At the same time, right, we can bring bicarbonate into the blood to buffer. So here's your favorite equation. Realize that in our cells, in these DCT tubule cells, we make carbonic acid, and then we have the ability to dissociate that, right, into hydrogen and bicarbonate. And so what we're showing here is the secretion of hydrogen, the reabsorption of bicarbonate into the bloodstream where it acts as a buffer. So those two things together definitely help neutralize pH. The other thing that happens here in these distal convoluted tubule cells is the deamination of amino acids. So again, as we're thinking about secretion, we're trying to get rid of waste products. So in that deamination process, one of the byproducts of that is ammonia. And we can go ahead and just secrete that into the urine. Remember, we're doing deamination, we're removing that amine group essentially to turn amino acids into some sort of fuel source. Another kind of nifty byproduct of that is through that deamination we also make more bicarbonate. We increase the buffers essentially in the bloodstream as a byproduct. Don't worry too much about that one, right? But we are interested in the fact that part of this secretion is removing waste products. So while reabsorption does happen at the DCT, we do tend to think more about secretion because we haven't really talked about it yet. Acids being secreted, waste products being secreted, drugs, toxins, etc. are also going to be sent to the urine at this point. Look at this, we're almost there. We are to the collecting duct, or the collecting system here. Remember, this collecting duct, not technically part of the nephron, because we actually have lots of nephrons coming in here, right? Lots of distal convoluted tubules dumping into the same collecting duct that actually multiple collecting ducts dump into what's called the papillary duct, and then we're delivering this to what's called the minor calyx. We basically now have urine that's in the kidney and is heading for the bladder. But notice what we're doing here. We have variable reabsorption of water and these ions. So let's look first at this reabsorption, sorry, and there is secretion, we'll talk about this. This idea of variable reabsorption of water. I really think of the collecting duct and the DCT as very similar because we have this variable reabsorption. And the variable here is the hormonal inputs, things like aldosterone and antidiuretic hormone telling us how much to reabsorb. Simple cuboidal, even into columnar cells as we move along the collecting duct. Now, notice what happens here. They've got these two images that are going to try to show us the effects of antidiuretic hormone. So in this image, antidiuretic hormone is not involved, and in this one, we see antidiuretic hormone's influence on the collecting duct. So notice what they're showing us here is basically there are times when we want to get rid of lots and lots of water, and so we produce large volumes dilute fluid, dilute urine. You know this, right? You've had 3 cups of coffee this morning and you're just like running to the bathroom to get rid of it, right? A large volume of dilute urine. Notice what happens though when antidiuretic hormone gets involved. We start putting a lot more water channels, those aquaporins, into the cells lining the collecting duct, and we see all of this reabsorption of water, right? So when we made the filtrate, it was really dilute. We went through this period of kind of concentrating it and then pumping the sodium out. But remember that sodium and chloride sets up this concentration gradient. We have a dilute filtrate again, and if that's fine with the body, if we have plenty of water, we just go ahead and pee it out. When the don't pee hormone comes along, It says, no, now is time to use this concentration gradient. So as the filtrate starts passing through here, remember, water movement, osmosis, is a completely passive process. All antidiuretic hormone does is put more channels in to make it possible. But say we're at 300 milliosmol, as we start passing through here, if this is 300, we lose water, right? The filtrate becomes more concentrated, but so does this paratubular fluid, and so we can lose more water, right? 900, 1200, and now we can actually concentrate our urine. But again, only to about 1200 milliosmol. So in the collecting duct, definitely think variable reabsorption of things like water, sodium, etc. Right? So aldosterone does work here. I don't have another good picture. Aldosterone does work here as well and tells us to reabsorb sodium. The other thing that we're doing here is making more determinations on pH, right? So if you have a low pH, if you're acidic, right, we will go ahead and secrete that hydrogen and reabsorb bicarbonate, just like we did in the distal convoluted tubule. Less common, but if you have a high pH, and it's less common because a lot of your metabolic processes are actually producing acids, think like lactic acid. But if you do have a higher pH, we can go ahead and secrete bicarbonate and reabsorb hydrogen. So we will see that the kidney, particularly the DCT and the collecting duct, are really important in buffering blood pH. Right? Again, we're also talking about secretion here at the collecting duct, so various acids and bases, again, drugs, toxins, etc. can also be added here. Oh, I don't even want to tell you this, but I'm going to. One of the other things that we reabsorb is urea. And this makes no sense to me because I'm like, it's a waste product, we want to get rid of it. But it turns out that in this whole concentration gradient, we actually reabsorb urea into the medulla here and also kind of strengthen this concentration gradient. Obviously only to a point. We do want to get rid of urea as a waste product. Okay. So at the end then of the collecting duct, we have urine. I can quit calling it filtrate, I can now call it urine. And so just a quick little summary here, you will be working on this in lab, but notice like things like the pH is usually slightly acidic, right? This was our chance to dump some of those hydrogen ions. When urine test strips talk about specific gravity, this is similar to osmolarity. It's basically talking about the saltiness level of your urine, right? So it's kind of a weird term, but you know how, like in freshwater, you're not as buoyant as you are in salt water. So like in the Great Salt Lake or the Dead Sea that's really salty, you float much better. Specific gravity is saltiness, go with that. Right. We don't usually, on those test strips, we're not able to measure that osmolarity, but there you can see the relationship, the actual number of solute particles per liter, but that does change the buoyancy level as well, right? Most of your urine is really just water, right? We're making up to 2 liters, maybe more, right, depending how hydrated you are. Should be a light yellow. We know that can differ depending on how concentrated that we have to make it. Of course, sometimes it can smell like asparagus, and you really should not have bacteria in your urine. If you're working in the medical profession, right, you should be becoming familiar with this idea of collecting a sterile urine sample. So while urine in the bladder is more or less sterile, we shouldn't find bacteria in it. The urethra, that's not always the case. And so it is important to actually clean the urethra before, and you don't have to like go up inside the urethra and clean it, but really kind of cleaning external genitalia before collecting that urine sample so that we don't accidentally show up like there's a bacterial infection when there's not. Okay, so this really was the craziness that took place at the kidney, right? All the way back to that renal corpuscle. Remember, 20 to 25% of the cardiac output is coming to our kidney, and here's why: because we're filtering it. This does a nice job of showing that efferent arteriole, right, that's going to become a paratubular capillary, so that every time we're reabsorbing things, we know it's going back into the blood, right? Proximal convoluted tubule, reabsorption dominates. We're gonna regather most of those things. We see the nephron loop setting up this concentration gradient. Water's reabsorbed on the descending side, sodium and chloride reabsorbed on the ascending side, right? Our distal convoluted tubule, they're showing the DCT and the collecting duct, they're showing the role of hormones like aldosterone and ADH in regulating that reabsorption. So remembering those are important there as well. DCT and collecting duct is also where we see a lot of adjustments made to pH, so the variable reabsorption of hydrogen or bicarbonate. So it's actually a pretty cool process. You will never look or think about urine in the same way. All right, so once this urine is finalized in the kidney, again, the only other thing I see students messing up is confusing a ureter with the urethra, right? So those terms do sound really similar. Make sure you get that straight. You'll be working on a lot of this anatomy in lab. I would point out, so male and female differences in the urinary tract, a lot of it comes down to the difference in the length of the urethra. Females get more urinary tract infections because the urethra is shorter. It's much easier for bacteria to make their way up into the bladder and potentially really bad on up into the kidneys, right? If they've made it that far. The urethra is shorter. It's also in much closer proximity to bacteria, say from the large intestine. E. coli is the #1 bacteria that leads to urinary tract infections. And then the last physiology thing here is the micturition reflex. This is the urge to urinate. So this is reflexive when you get about 200 milliliters of urine in the bladder, right? We're actually going to start this reflex. Basically, the reflex is that you start contracting some of these detrusor muscles, so some of these muscles in the bladder, and that actually elevates the pressure, right? So you feel more like you have to urinate. Notice also, right, that we have a signal going to the brain. So you do get the message, hey, the bladder's full. Now you have two different sphincters involved here, right? Notice the blue dotted line from the brain is going to the external urethral sphincter. This is the one under voluntary control, right? Your brain decides when it's time to go. You do have an internal sphincter as well that's involuntary, okay? So let me show you this one. The interesting thing with the sphincters, it might be easier to see in the female anatomy here. Here's the internal sphincter, right at the opening of the bladder into the urethra. That's under involuntary control. So when the bladder fills, right, that can relax, but our external urethral sphincter under voluntary control can stay, right? Unless it can't. That would be incontinence if you don't have control over that external sphincter. This external sphincter is located in a band of muscle at the bottom of the pelvic cavity. This is called the urogenital diaphragm, like a band of muscle, and it's embedded in there. So if that diaphragm is strong, right, that supports the external urethral sphincter and your ability to have voluntary control over it. One of the most common ways that diaphragm is weakened is through childbirth. I mean, think about it, right? What kind of trauma is taking place there to that musculature? And so it can take a while to regain the voluntary control over that. Kegel exercises can be useful in strengthening the diaphragm there, because it is nice to be continent and decide when you are going to micturate, when you're going to